Hello everyone, my name is Coline Gianfrotta and I represent a study entitled on the predictability of A-minor motifs from their local context in RNA structures that has been published last year in the journal RNA Biology. So, some recurrent substructures appearing frequently in RNA structures called motifs remain unpredictable. This is the case of the type 1,2 A-minor motif that is an important and widely represented 3D motif. It connects a loop of secondary structure to an helix through non-canonical interactions. We will focus on this motif in our study and particularly on finding what other structural information we could provide to help to predict this motif. And so the purpose of our approach is to study the structural context of A-minor motifs, which means the structure around an A-minor motif, in order to determine what kind of information contained in the structural context can be useful to characterize and predict these motifs. And so we studied the structural context at two scales, the topological context, which is composed of the interactions around the motif, and the 3D context, which is the local 3D substructure around the motif. And first of all, we will rapidly see how to use 3D context to classify A-minor motifs. So, we define the 3D context of an A-minor motif as the 3D substructure, so the coordinates of all atoms, containing the motif, and 3 nucleotides in an orbit around the motif on the sequence. So we have here two examples of 3D context. And we then compare two 3D contexts by aligning them nucleotide by nucleotide and by calculating the RMSD of the alignment to obtain a measure of similarity between the two structures. And finally, we classify our motifs according to their 3D context similarity by using a clustering algorithm on a graph like this, where one vertex corresponds to one A-minor motif and there is an edge between two vertices if the RMSD between them is lower than the threshold that we chose experimentally, and here it is 2.5 angstrom. So you can find more details about the classification on the poster actually. And we will now see how to use this classification for prediction. So first we define the topological context of an A-minor motif as a set of covalent, canonical and non-canonical interactions around an A-minor motif. And by around we mean at a distance lower than 3 nucleotides on the sequence. And we represent this topological context as a directed graph where the vertices are the nucleotides and the arcs, so the links, are the interactions. So we have an example here. We consider 3 nucleotides on the sequence, like we did for 3D context, and also all the interactions involving these nucleotides. And after defining this topological context, we will see how to use it in our study. So if we take our 3D classification, we can construct the topological context for every motif of every class. So we have here, for example, the topological context of this particular class. And we will consider the maximum common subgraph between all the topological context of one class, which means basically the interactions that they have in common, for example here. And we thus can define the topological representative for every class, which is the maximum common subgraph without the interactions of the motif. And secondly, we define sequence representatives as well. So it is two sets of regular expressions, one for the loop strand of the motif and one for the helix strand. And they correspond to the union of all possible sequences of context in the class, induced by the maximum common subgraph, which means that the type of nucleotide is considered only when the nucleotide is in the common subgraph. And the question we want to answer is, can the presence and the position of A-minor motif be inferred from these representatives? And to answer this question, on one hand, we use a dataset of 374 A-minor motifs from the database Carnaval in November 2018, and we construct the 3D classification with this dataset. And on the other hand, we took all the non-redundant structure of PDB in June 2020, and we separate them into two sets. One validation set in which there are the structures containing the A-minor motifs we use to construct our classification, and a test set with all the other structures. And in these sequences and structure, we search for our class representatives for every class containing more than 3 A-minor motifs. And to evaluate our result, we use two measures. So the first one is a PPV, positive predictive value, one per class, and we calculate one PPV for the loop strand and one PPV for the helix strand, because we have one set of regular expressions per strand, and the topological representative can also be considered in two parts, as we can see here, for example. So I will present you the results of PPV values obtained by using the two types of representatives, so the topology and the sequence. So we have here the class number on the x-axis and 4 values of PPV for each one of them, 2 for the validation set and 2 for the test set for loop strand and helix strand. And what we can see is that some classes have very low values of PPV. This is particularly true of the classes in red here, that are the classes containing non-homologous motifs, and some of the other classes have PPV values greater than 0.7 for both datasets. So these classes are quite predictable, this one, and all the other classes have lower PPV value, especially for the test set. And it gives average values around 0.5 for the validation set and around 0.3 for the test set. So we have a good signal for some particular classes that contain only homologous motifs. And the second predictability measure we calculate is the TPR, so the true positive rate for all classes. And we have around 80% of A minor motifs that are found with the two representatives in the validation set, which is expected to be high since the majority of the motifs of this dataset are in the classes we use. But we also have a quite high amount of motifs found in the test set, with 60% of new motifs found with our two representatives. So, in conclusion, we can say that we developed an automatic graph-based method to classify A minor motifs according to their 3D context similarities, and we computed sequence and topological representative for every class to determine whether these representatives can help to predict the presence and the position of an A minor motif. And we found new subclasses of A minor motifs according to their 3D context similarities with our 3D classification. And for prediction, for particular A minor motifs, topology combined with a sequence signal seems to be sufficient to predict their presence, but in most other cases, topological and sequence information is not sufficient for prediction, but it could be a good marker anyway. Perspectives for prediction, so to try to have better results for prediction, we could change our graph model to be closer to 3D structures, for example, or maybe calculate less deterministic topological representatives in order to find more true positives. And in a larger point of view, we could see our class representatives as meta-motifs, so bigger motifs that may be used in supplementary input to existing prediction methods, for example. And so, thank you for your attention.